Okay, today we are going to discuss the second example related to the internal structure of storage roots. The first one we discussed was beetroot, and that was the taproot type, and now the second one is sweet potato, which is a tuberous adventitious root. The botanical name of sweet potato is Ipomia betatus, and it belongs to family Convolvulaceae. The primary structure is just like beetroot, but there we draw the diarch condition of xylem, but here it can be penta to hexaarch. Then there is development of vascular cambium, that part is also same, but the activity of vascular cambium is anomalous. Okay. So, in this case, adventitious fleshy root is formed by anomalous secondary growth. So, the secondary growth is anomalous here as well. Cambium is normal, but its activity is abnormal. Okay, so first thing about its primary structure. Primary root is pentarch to hexarch. It means 5 to 6 patches of phloem alternate with xylem. Pith is absent. Abundant cortex. Epidermis with unicellular hair. Endodermis and pericycle are present. So, this is the primary structure. Now, during secondary growth, the primary cambium means the first formed cambial ring, which is also called vascular cambium. So, the primary cambium develops just like normal dicot root, and it cuts secondary xylem towards inner side and secondary phloem towards outer side. Secondary xylem has few vessels. Most of parenchyma to store food. Okay, so generally we see that xylem vessel and trachea elements are abundant and parenchyma is lesser in the vascular region, but here the vessels are very few and majority of the secondary xylem includes parenchymatous cells which store food. Later on, it separates vascular strands in ground tissue. Okay, so the few elements, the vessel elements from the secondary xylem form towards inner side, they get separated from it and they come to lie in the ground tissue area. Okay, now the anomalous growth will start. The secondary cambia develop around individual vessels or vessel groups. These cut new xylem elements towards the vessels and few sieve tubes and latex canals away from vessels, means outside the cambium. Okay, so here we will see that some of the xylem vessels which got separated from secondary xylem and which are lying on ground tissue, they develop the second type of cambia called secondary cambia. Now these secondary cambia, they are like a ring around a single vascular element or more than one vascular elements, and their activity is that they will cut more vessels, more xylem elements towards the center and few sieve tubes and latex canals towards the outer side. So it is an anomalous type of secondary growth. Large amount of parenchyma is formed in between. So this was the activity of secondary cambium. So initially we had primary cambium, then secondary cambium. Now there will be the formation of tertiary cambium. Okay. Tertiary cambium develop later on from storage parenchyma the epidermis gets ruptured and lignified. Cortex cells divide to fill the gap due to secondary growth inside, because the elements are developing, secondary xylem, secondary phloem, lot of storage parenchyma. So because of that, the diameter of the root is increasing. Now it will pose stress to the immediate cells, that is endodermis, cortex and epidermis. The endodermis survives for some time, its cells get stretched, but later on it disintegrates, but it is visible at the earlier stages. whereas the next layer, cortex, its cells, they start dividing because they are facing pressure. So, they will start dividing in order to fill the gaps which are being created because of the increased diameter, because of the increased laying of secondary tissues in the center of the root. Okay. Now, the epidermis, that may either get slowed off completely so that it is disintegrated, or it may get ruptured at few places and lignified. So, that is how the changes in the primary tissues take place because of secondary growth. Okay, the endodermis cells are stretched and disintegrate due to pressure developing from secondary tissues towards the outer side coccambium develops from pericycle and forms coc and secondary cortex to form periderm. Okay, this part is same. The third step is same, that is the formation of periderm. So, now we are going to draw diagrams. We will draw only two diagrams in this case. One is related to the primary structure and the second one is for the secondary growth, a fully developed root showing secondary growth. So, for the primary structure, we will show stellar region, so draw a circle just slightly bigger, a dotted layer for showing pericycle, another circle again close to it, slightly bigger, the endodermis, okay, and leaving some gap for cortex, the epidermis. Okay, so first we are going to draw primary xylem. So, in primary xylem we have large elements in the center. You can draw 2 or 3 very large elements, and afterwards the condition is pentarch to hexarch, means you can show 5 or 6 branches of this primary xylem. So, I am going to draw here 5 branches, it is a pentarch condition. So, along these lines you will make hexagonal cells or polygonal cells, may show branching at certain points. They may be slightly out of the line, okay. It is not necessary to make perfectly straight elements because they are not so in the original root. So, this is primary xylem. So, as we have already discussed, the xylem should be double walled, leaving a wide lumen. Okay, so this is primary xylem, and the alternate patches would be of primary phloem. So in these alternate areas, again the size may vary. And the cells of phloem should be small polygonal cells So we will complete one area only, and in between these are the cells of conjunctive parenchyma. So you can draw here polygonal, slightly elongated cells. So you have to fill all these gaps. Make them concentric around phloem. Afterwards you can fill the gap with remaining cells. Okay, so on you have to complete this part. Towards outer side we have the pericycle and then endodermis. So pericycle, these two lines I am using to draw pericycle. You are not to draw just the cross sections between the lines. Mark proper rectangular cells. Okay, don't show like this, just the cross sections. So this is pericycle. Outside pericycle is endodermis. So endodermal cells larger than pericycle, barrel shaped, means their outer wall should be slightly bulged. Then outside we have the cortex and epidermis. So epidermis, single layered rectangular cells without intercellular spaces. You have to complete this layer. This is the outermost one. So you need to draw all cells. Okay. And in between this zone there is present cortex. So the cortical cells are, you can show polygonal cells as well, because in this a specific pattern is observed. We have large cells, small cells abundant with a kind of round polygonal dimensions. So it is your wish, you can show polygonal cells as well as circular cells. So fill the gap, showing variation of sizes, variation of orientation, shape. So we'll fill few cells only. Okay, and abundant root hairs are present on the surface. So you will firstly erase the cross section and then make the tubular extensions of epidermal cells as root hairs. These hairs should be cylindrical and their tips should be round. Okay, you can have variable lengths, but the tips should be round. This way you will cover the entire surface. So the labeling part, we have root hair, epidermis, cortex, endodermis, pericycle, primary phloem, primary xylem, and in between these we have, within this space between xylem and phloem, there is conjunctive parenchyma or conjunctive tissue. So this is TS of primary structure of sweet potato. So now